Okay, hello. This is the lecture material for Chapter 12, Microscopic Eukaryotes, and this is Part 1. You can see I'm using a new recording setup. You'll be able to see me work through the slides, and then if there's anything important I want to show you, I can do that too. So let's get on with it. Lecture 16, Chapter 12, Eukaryotes 1. That's what I intend to get through and post for you today, which is Friday. So, an introduction to the types of microscopic eukaryotes, a look at the difference between a heterotroph and an autotroph, look at the difference between eukaryotes and prokaryotes, and then in this first section also looking at the fungi and their structure and classification. So looking at slide 2, you can see that we have various things that are eukaryotes that we could include in microorganisms, i.e. in organisms that we need to see, that are very small, that we can see through a microscope. So the first thing is, think back to our domains. We have our bacteria, we have our archaea, and we have our eukaryotes. And these are three branches of these three domains. I'll put this in front. There we go. These are all as different from each other as they are from every other one. So the archaea are as different from the bacteria as the bacteria are from the eukaryotes, and the bacteria are as different to the eukaryotes as the eukaryotes are to the archaea. Now in our eukaryotes, if we try and superimpose, remember our 6-kingdom system, we have our bacteria, our archaea, and then we have our eukaryotic kingdoms. Things like plants, fungi, animals, and this group of stuff called protists. So we are only going to deal with the things that are eukaryotes in this set of lectures. So if we were to write out our 6-kingdom system, we have our two, what you might call prokaryotic kingdoms, or indeed our two domains, and our third domain here is split up into 4 pieces. And the first problem with the 6-kingdom system, and I mentioned this problem before, is that idea that not all of these kingdoms are equal. These 2 kingdoms are domains, whereas these 4 eukaryotic kingdoms are all part of the same domain. But one of the real big problems with the 6-kingdom system is the protists, because it used to be the case that you would put things into classification based upon what they look like, where they lived, and so we can quite easily think about things that are plants. And we can quite easily think about things that are fungi. And we can quite easily think about things that are animals. And then there was all this other stuff that really it wasn't really much like a plant. or at least physically, appearance-wise. It didn't really fit in the fungi, it didn't really fit in the animals. So all of these disparate organisms were lumped together in the protists. And they don't belong together. There are members of the protists that should be here in the animals, because when we look at their evolutionary relatedness, we see that they are the green algae, for example, and plants share a common ancestor. There are organisms with common ancestors of animals and some of the protists. There are common ancestors with the fungi and some of the protists. So that goes to our slide here, our second slide. The fungi, those are quite a nice defined group, but the protists are not. And that's why it says here, informal groups. The protists is a very informal group. If you were working in taxonomy or classification right now, you would not be using the term protist to define a group of organisms. Within the protists, we're going to look at, so I'll use the term protist just because that is it's a term that kind of denotes simple organisms, either single-celled organisms or organisms that have a simple structure. One type of protist is the algae. These are photosynthetic eukaryotes. Another kind of protist are protozoans, and these are microscopic heterotrophs. So, what is a eukaryote? What do these informal groups mean? That should be enough to cover that. Let's look at this difference between heterotrophs and autotrophs. Autotrophs, auto means self, troph means feeding, self-feeding organisms, and if you are a eukaryote, pretty much there's only two ways you can get your energy, being an autotroph or being a heterotroph. Also two ways of getting your carbon. If you are an autotroph, you are a photosynthesizer. That means you perform photosynthesis and you harvest sunlight energy, and you put that energy into the bonds of molecules such as glucose. And since our autotrophs in this case are eukaryotic, they have inside their cells organelles, and one of those organelles is the chloroplast, and inside the chloroplast is that chemical chlorophyll, and it's chlorophyll that allows the autotrophs to capture the sunlight energy. The other way you can make your living is by being a heterotroph, and that means you ingest material from the environment. So humans are heterotrophs, we eat food, and then we break down the food, and then we extract the energy. Fungi are all heterotrophs. They don't eat per se, they have no mouths. What they do is they grow on their food source, they release enzymes, and then they reabsorb material. So the kinds of heterotrophs and autotrophs we're looking at: fungi are heterotrophs, the algae in the protists are autotrophs, the protozoans in the protists are heterotrophs. That protozoa means first animal. They have many animal-like qualities. And then also, in addition to these stuff here, we are going to look at multicellular organisms. In particular, we're going to look at the animals. And we're going to look at the wormy-like animals. So these aren't worms like you find in the garden. These are not earthworms. There are lots of different wormy-like animals. They just all share the same kind of long, tube-like body. Okay, so I was talking about the wormy type of animals. These are animals, so they're multicellular, and we'll look at their cell structure. And then the other kind of important animal we'll look at are the arthropods. Arthropods, things like insects and arachnids. Very important, not just because some of them do directly cause disease, but they're very important in spreading disease. They are vectors for many kinds of diseases. Okay, on to slide number 3. We've looked at the differences between prokaryotes and eukaryotes before, so I won't go into too much detail. One has a nucleus, the other one doesn't. One has membrane-bound organelles. Bacteria have peptidoglycan. No eukaryotes have peptidoglycan. But a couple of things we haven't mentioned. First of all, a cytoskeleton. So inside your cells, there are all of these tube-like elements that are giving your cells shape. And these tube elements, things like, they're called microtubules, they are made of proteins. Proteins arranged in a kind of a tube-like configuration. Not a particularly great drawing, but there we go. Only eukaryotes are known to have microtubules in their cells. Now, you may have heard of microtubules before in regard to cell division, mitosis and meiosis. It's microtubules that come in and grab hold of the chromosomes and then pull the chromosomes apart. We've talked too about the idea of chromosomes. Chromosomes in bacteria, they have that single circular chromosome. Even though they do have those additional DNA elements called plasmids, currently there are no bacteria, or indeed archaea, that have more than one chromosome. Those plasmids don't count as a chromosome. Eukaryotes have linear chromosomes. So for example, in a human cell, we have 46 linear chromosomes out of 23 pairs. Another term you've almost certainly heard before is diploid and haploid. So let's look at that human cell. Human cell is a eukaryote. It has a nucleus, and inside that nucleus there are all of the chromosomes. This would be a human body cell. I'll just represent a few chromosomes in here. Let me zoom in a little bit. So, diploid, di meaning two. So a diploid cell has two of each chromosome. And you may be familiar that you get one set of chromosomes from your mother and one set of chromosomes from your father. So whilst we do have 46 chromosomes, it's best to think of them as 23 pairs of chromosomes. This is the diploid cell, with two of each. And then when we go through a process such as meiosis, we end up with cells that only have one of each of those chromosome pairs. So you have half the number of chromosomes as the parent cell. They are haploid. And haploid cells are essential for sexual reproduction, because then two haploid cells can get together and they can fuse and they can recombine their genetic material to produce an offspring. When humans make haploid cells through meiosis, we call these cells gametes. But not always gametes are the production of meiosis. We'll see in fungi that spores are produced. Also in plants we have the production of spores. But we're talking about plants today. So sexual reproduction requires meiosis. To reduce the number of chromosomes so that those chromosomes can be recombined in the next generation. Asexual reproduction, a reproduction without genetic recombination, that can happen too. That can happen through in eukaryotes. It can occur via mitosis. So when I damage my skin, for example, mitosis occurs to fill in the gaps. But if you're a single cell, you can go through reproduction, i.e. parent and then offspring, through the process of mitosis. So on the slide here, there's a picture of a life cycle showing that various things can happen. A human being has a very simple life cycle. We have a long period of being diploid, and most of our lives we are big diploid organisms. We have a small part of our life cycle where we are haploid gametes, and then those haploid gametes get together and fertilize, and we're back to the diploid configuration. But there are some other things that can occur. You can have an entire multicellular organism that is entirely haploid. This is one configuration very often seen in the fungi. You can have haploid cells go through mitosis to produce more haploid cells. You can have mitosis of a diploid cell, making more diploid cells. And then you can have meiosis of a diploid cell, producing haploid cells. So lots of different organisms have more complex life cycles than humans do. And some of those life cycles involve multiple stages. And sometimes those life cycle stages, you can have a haploid life cycle stage separate to the diploid life cycle stage. In the case of fungi, you can have both of those stages can be multicellular, not just sex cells like we think of gametes. Bacteria and archaea, prokaryotes. Now, even though we have seen them get together and pass information, pass plasmids between one another, and even we call that structure that they use to pass the plasmids a sex pilus, all prokaryotes divide themselves via binary fission, which is a form of asexual reproduction. There is no genetic recombination in a bacteria or an archaea, at least not regarding the chromosome. Okay, I think I'll stop it there. That's the introduction of this first section here, and we'll pick up all those points when we come back later on.